So the next speaker is Laura Nicolucci, and she's going to talk about investigating the effect on targeting TRAF1 PKN1 signaling axis in inflammation. Welcome, Laura, and please share your presentation. Yeah, perfect.
Okay, thank you for that introduction. Hi, everyone. My name is Laura, and I am currently working at York University. And today I'm going to discuss my project, which is targeting the TRAF1 PKN1 signaling axis in inflammation. So inflammation is a natural and essential response of the immune system to injury and infection or harmful stimuli, and it helps the body fight off pathogens and initiate healing. However, this process needs to be carefully regulated to maintain a healthy immune response. Now, in an ideal scenario, there's typically a balance between pro and anti-inflammatory signals, and this ensures that the immune system responds effectively without causing unnecessary bodily damage. However, when this balance is disrupted, excessive inflammation can occur, and this prolonged and uncontrolled inflammatory state can contribute to the development of several types of chronic diseases, such as rheumatoid arthritis, cardiovascular disease, cancer, and many more. And something that is more alarming is that chronic diseases account for approximately 75% of all deaths worldwide. Now, in order to understand and develop treatments for these inflammatory conditions, we first need to explore the underlying biological mechanisms that are driving these conditions. And this involves studying key proteins and pathways that regulate this inflammation. Now, one of the proteins that is heavily focused on in our lab is TRAF1, otherwise known as TNFR-associated factor 1. And it is a signaling adapter protein, and it is present in both lymphocytes and monocytes. Now, in the context of TNFR signaling, downstream of CD40, TRAF1 recruits CIAP1 and 2, which stands for cellular inhibitors of apoptosis proteins, and they function as a dual ligase. Now, the recruitment of CIAP2 leads to the addition of K63-linked ubiquitin to the TAC-TAB complex, thereby activating TAC1 and leading to the activation of MAPK and NF-kappa B pathways, promoting excess survival signaling. Now, under typical conditions, the TRAF1-CAP2 interaction results in the addition of K48-linked ubiquitin to TRAF1, marking it for proteasomal degradation. However, it has been shown that TRAF1 phosphorylation by PKN1, a member of the protein kinase C family, prevents this degradation of TRAF1 by stabilizing it and leading to sustained and enhanced pro-survival signaling. Now, in collaboration with the Watts Lab at the University of Toronto, we investigated the role of PKN1 phosphorylation in regulating TRAF1 stability. Now, using OTSSP167, a known MELK inhibitor, which is a maternal embryonic leucine kinase that's overexpressed in breast cancer, we inhibited PKN1 phosphorylation in lymphoma models, and results show that inhibiting this phosphorylation led to the destabilization and loss of TRAF1, leading to lower pro-survival signaling, and further proving that PKN1 is necessary for TRAF1 stability downstream of CD40 signaling. Now, at the time of that study, there was no direct way to essentially measure phosphorylated TRAF1, or phospho-TRAF1, as I'll probably refer to it for the rest of the presentation. So again, in collaboration with the Watts Lab, along with Abcam, we actually developed a phospho-TRAF1 specific antibody to track the changes in phospho-TRAF1 to better understand the phosphorylation dynamics. Now, to validate our antibody and to confirm the loss of phospho-TRAF1, in panel A, we essentially treated 293 T cells with varying doses of the PKN1 inhibitor, OTSSP167. So the concentrations range from 10 to 100 nanomolars, and higher doses led to the loss of both phosphorylated TRAF1 and TRAF1, and similar results are shown in panel B in actual patient samples with chronic lymphocytic leukemia, confirming that the loss of phosphorylation reduces both phospho-TRAF1 and TRAF1 levels. So now in a previous and more recent project conducted by our lab, the role of the TRAF1 CIAP2 axis in innate immune cells was investigated to determine its involvement in inflammatory signaling and inflammation rather than survival signaling, as we saw in the TNFR signaling complex downstream of CD40. So their work revealed that similar to CD40 signaling, TLR signaling in monocytes also involved TRAF1, and this complex recruited CIEP1 and 2, which led to the enhancement of MAPK and NF-kappa B signaling, contributing to inflammation. Furthermore, a point mutation in TRAF1 at position V203A disrupted this interaction with CIEP2, resulting in an overall reduced inflammatory gene expression and signaling. Now, while inducing mutations or creating genetic knockouts can provide valuable insights, these approaches are not necessarily ideal for therapeutic development due to their complexity and lack of specificity in clinical settings. So this led us to question, what is exactly causing the stabilization of the TRAF1 CIEP2 complex, and whether we can target this mechanism to destabilize TRAF1 and prevent its degradation from CIEP2. Given this, we hypothesized that PKN1 might be playing a critical role in stabilizing the TRAF1 CAP2 complex by phosphorylating TRAF1 within the TLR4 complex. If this phosphorylation were responsible for TRAF1 stability, could targeting this modification disrupt this interaction, leading to TRAF1 destabilization via CIAP-mediated degradation and ultimately reduce the inflammatory response? So essentially, the goal of our study is to investigate the role of PKN1 in TRAF1 stability within the TLR signaling complex to reduce inflammatory signaling. To do this, we first generated PKN1 knockouts in THP1 monocytic cell lines using CRISPR-Cas9, and we assessed the impacts of PKN1 knockout on inflammatory gene expression and inflammatory signaling. And as I previously mentioned, while genetic knockouts provide some sort of insight into PKN1's role in phosphorylating TRAF1 downstream of TLR signaling, it's not necessarily a viable therapeutic approach. Now, for a more therapeutic approach, we turned to OTSSP-167, and kinase screening that was previously done revealed that OTSSP-167 was the most potent in reducing TRAF1 levels, which is why we decided to use it for our experiments. So we then treated wild-type THP1 cells with varying concentrations of the inhibitor and examined its effects on inflammatory gene expression and signaling. Okay, so in the first panel up here, we performed a Western blot analysis on THP1 cells stimulated with LPS for 24 hours, and they were also simultaneously each treated with varying concentrations of OTSSP-167. So as shown in figure 1, we observed a dose-dependent reduction in both TRAF1 and phosphorylated TRAF1 levels at higher concentrations of OTSSP-167. And this result essentially provides kind of the first indication that TRAF1 is phosphorylated in monocytes. Statistical analysis of these findings confirmed that the reduction in both TRAF1 and phospho-TRAF1 levels was statistically significant, further supporting the role of PKN1 phosphorylation in stabilizing TRAF1 in these cell lines. Now, to assess the effects of OTSSP-167 on inflammatory gene expression, we performed qPCR on wild-type THP1 cells stimulated with LPS for 1, 3, and 6 hours. We focused on key pro-inflammatory genes, including TNF-alpha, IL-1 beta, and IL-6, as these cytokines are crucial mediators of the inflammatory response, and especially in monocytes, and play a key role in driving both acute and chronic inflammation. So as shown in this second figure here, we observed a reduction in the expression of all 3 inflammatory genes over the 6-hour time course. Now, while this data provides preliminary evidence that OTSSP-167 inhibits inflammatory gene expression, further experiments are needed to confirm these results and establish statistical significance. So now we're moving on to my PKN1 genetic knockouts. So to assess the effects of PKN1 knockout on inflammatory signaling, we performed flow cytometry on wild-type and PKN1 knockout THP1 cells stimulated with LPS for 15, 30, and 60 minutes. We measured the phosphorylation of key signaling molecules, both phospho-P65 and phospho-ERK, as these markers are critical for the activation of NF-kappa B and MAPK signaling pathways, both of which are essential for the inflammatory response. So as shown throughout this entire figure, at time points 15, 30, and 60, we see a significant reduction in both the phosphorylation of phospho-P65 and phospho-ERK in the PKN1 knockout cells compared to the wild-type THP1 cells. So these results suggest that the absence of PKN1 leads to impaired activation of key inflammatory signaling pathways. Okay, next, focusing on the top panel here. So we wanted to investigate the effect of PKN1 knockout on inflammatory signaling, and we performed a Western blot to assess phospho-TAC1, phospho-P65, and phospho-Jun. Now, just as a brief summary, so phosphorylated TAC1 activates both the MAP kinase and the NF-kappa B pathways. So focusing on MAP kinase, phosphorylated TAC1 will eventually lead to Jun phosphorylation, leading to the activation of AP1, which regulates pro-inflammatory gene expression. And additionally, TAC1 also activates phosphorylated P65, which drives inflammatory gene expression or transcription through activation of NF-kappa B. So looking at panel A, we assessed phospho-TAC1, phospho-P65, and phospho-Jun phosphorylation in wild-type and PKN1 knockout THP1 cells treated with LPS for 15, 30, and 60 minutes. Now, we observed a significant decrease in both phospho-P65 and phospho-TAC1 levels in the knockout cells. While phospho-Jun results are still preliminary, a downward trend is still observed, and these findings suggest that PKN1 plays a role or a key role in regulating inflammatory signaling pathways, highlighting its potential as a therapeutic for modulating inflammation. And now to looking at panel B, so to further investigate the role of PKN1 in inflammatory responses, we performed qPCR to assess the expression of key inflammatory genes. Similar to what we used for the inhibitor experiment, we looked at TNF-alpha, IL-6, and IL-1 beta, which are crucial in monocyte-mediated inflammation. So the cells were stimulated with LPS for 1, 3, and 6 hours, and we observed statistically significant differences in gene expression between the PKN1 knockout clone and the wild-type THP1 cells. Now, these findings provide further evidence that PKN1 plays an essential role in modulating inflammatory gene expression and highlights its potential as a target for controlling inflammation. So currently I am working on an immunoprecipitation experiment where I am pulling down TRAF1 with the CIP2 complex to ensure that phosphorylated TRAF1 is being lost from the CIP2 complex when the inhibitor is added, as well as in the PKN1 knockouts. So this will just essentially help confirm the localization of TRAF1 phosphorylation within the MyD80 dependent signaling complex at the TLR4 membrane, in line with our hypothesis that TRAF1 is phosphorylated by PKN1 within this complex. Now, building on this study, future work will focus on the development of a more potent and specific inhibitor of PKN1, and this could be tested in mouse models to evaluate its effects on inflammation, with the ultimate goal of advancing it to clinical trials for individuals with inflammatory diseases, offering a more targeted therapeutic approach to reduce overall inflammation. Now, I would just like to thank my amazing supervisor as well as my lab for, constantly being very supportive of me throughout this project. And I would also like to give a special thanks to the Watts lab for your collaborative efforts during this project as well. So thank you.